Hey everyone, let's get right into it. Today, we're diving into Core Paper 2, Genetics and Molecular Biology. Think of this explainer as a journey to uncover the ultimate source code of life. We are moving way beyond dry textbook facts here, because we want to truly understand the magnificent microscopic blueprint that literally makes you, you. We're going to break down the core principles of genetics, exploring exactly how biological information is stored, how it's transmitted, and ultimately how it's brought to life.
It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This famously wry, understated little sentence from James Watson and Francis Crick back in 1953 quite literally changed biology forever. With just as one incredible insight, the deep mystery and the profound elegance of genetics began to unravel. It shifted our entire understanding of heredity from some abstract fuzzy concept into tangible concrete chemistry.
Okay, let's dive into this. Here is our roadmap for today. First, the search for heredity. Second, unlocking the double helix. Third, the central dogma. And finally, regulation and mutation.
Starting with Section 1, the search for heredity, finding the blueprint.
Figuring out the exact molecules of inheritance was seriously one of the greatest scientific detective stories of the 20th century. For a very long time, basically right up until the 1940s, scientists mistakenly thought proteins were the genetic material. And honestly, you can't really blame them. Proteins are incredibly complex and diverse, which naturally seemed necessary if you're trying to carry the blueprint for all of life's vast diversity, right? Nucleic acids, on the other hand, just seemed way too simple. But a rapid-fire sequence of discoveries totally flipped this assumption on its head, starting back in 1928, building through 1944, and culminating with definitive proof in 1952.
The whole breakthrough started with a British medical officer named Frederick Griffith. While he was trying to create a pneumonia vaccine, he did something fascinating. He mixed dead, heat-killed pathogenic bacteria with live, totally harmless ones. Then, he injected that mixture into mice. Shockingly, the mice died. Somehow, the live, harmless bacteria had turned deadly. Griffith realized that a physical, heritable trait could actually be transferred between cells, a process he called transformation.
And this brilliantly illustrates the final nail in the coffin for the whole protein theory. In 1952, Alfred Hershey and Martha Chase used bacteriophages, which are basically just viruses that infect bacteria. They brilliantly used radioactive isotopes to tag the virus's protein in one batch and its DNA in another. What they found was that when the virus hijacked a host cell to reproduce, only the radioactive DNA actually entered the cell. The DNA, not the protein, was the thing doing the programming.
So why does all this matter to you? Well, the discovery of transformation wasn't just some quirky lab observation about mice and bacteria. It proved once and for all that a specific chemical molecule dictates your physical traits. It established the chemical basis of life itself, giving us the fundamental proof that your genetic endowment, again, the very stuff that makes you you, is a real, physical, transferable substance.
All right, let's move on to Section 2, unlocking the double helix. The structure of code.
Now that science finally knew what the genetic material was, the next massive hurdle was figuring out how in the world this molecule's physical structure could possibly store so much complex data. A huge clue came from biochemist Erwin Chargaff. He made a really peculiar observation about the 4 nitrogenous bases in DNA. He found that across different species, the amount of adenine always perfectly equaled the amount of thymine, and guanine always perfectly equaled cytosine. These became known as Chargaff's rules, and they were the absolute crucial puzzle piece needed to finally crack the DNA code.
By putting all these clues together, and relying on some absolutely crucial X-ray diffraction data captured by Rosalind Franklin, Watson and Crick built their iconic model, the double helix. It features 2 anti-parallel sugar phosphate backbones winding around the outside, with those nitrogenous bases, the A's, T's, G's, and C's, paired up beautifully on the inside, holding the whole structure together just like the rungs of a twisted ladder.
Now, what's really interesting about this is the backbone itself. You have phosphodiester linkages securely connecting the sugars and phosphates, but pay attention to those bases projecting inward. Because of strict base pairing rules, A only ever bonds with T and G only with C. However, and this is the magic part, there is zero restriction on the linear sequence of these bases along a single strand. That linear sequence can vary in countless, basically infinite ways, which allows a simple 4-letter alphabet to code for literally every unique gene in your entire body.
But you might be wondering, why DNA and not its close cousin, RNA?
Well, it all comes down to chemistry. RNA has a highly reactive 2' hydroxyl group on its ribose sugar, which makes it chemically unstable and easily degradable. DNA stands for deoxyribose, meaning it's actually missing that oxygen. That tiny difference makes it chemically far less reactive and structurally incredibly stable. Basically, DNA is the perfect, secure, long-term hard drive for our source code, while RNA's temporary, fragile nature makes it the absolute perfect short-term messenger.
Which brings us to Section 3, the central dogma, the information highway.
Okay, so we have this highly stable double helical hard drive, but how on earth does a cell actually read it? The answer lies in a fundamental concept proposed by Francis Crick called the central dogma of molecular biology. This dictates the one-way, directional flow of genetic information, going from DNA to RNA to protein. You can essentially think of this as the universal operating system for almost all life on our planet.
When you follow this pathway, you really start to see the sheer genius of the system. The master code, the DNA, stays locked up and protected inside the nucleus. When a specific gene needs to be expressed, that segment of DNA is transcribed into a temporary RNA messenger. That messenger then leaves the safety of the nucleus and is translated into a protein. And proteins are the actual molecular machines that do the real, physical work in our cells.
So to break it down, we have 3 core processes keeping you alive right this very second. First, replication, which ensures identical copies of the DNA are made every time a cell divides. Second, transcription, which safely copies a gene into an mRNA messenger without risking the original DNA master copy. And third, translation, which reads that messenger to build the physical proteins your body needs to actually function.
Let's move to and see how this builds during the replication phase. Because of that double helix structure we talked about, replication is what we call semi-conservative. The 2 parent strands literally unzip at a replication fork, and each single strand acts as a precise complementary template. New nucleotides line up along each template, ensuring the brand new double helix is an exact, perfect replica of the original.
Then, further down the line, during translation, that chemical genetic data becomes physical reality. A cellular machine called a ribosome reads the single-stranded messenger RNA in specific sets of 3 letters, which we call codons. Each 3-letter codon translates perfectly to one specific amino acid. The ribosome just keeps reading, stringing these amino acids together like beads on a necklace, and eventually folding them into those complex, functional proteins.
Finally, we reach Section 4, regulation and mutation, controlling the system.
We've seen just how vast and intricate this genetic machinery really is. But remember, it exists in a harsh, dynamic environment. So what actually happens when the genetic code changes or gets damaged?
The harsh reality is our DNA is under constant, daily assault. Physical and chemical mutagens, like ultraviolet light from the sun, can fuse adjacent bases together into these things called dimers. Reactive chemicals can cause alkylation or oxidative lesions. Spontaneous chemical reactions can even cause depurination, where a base just drops right off the strand. We even face devastating double-strand breaks. It's a remarkably hostile environment.
So the crucial point is this. If our DNA is constantly being attacked like this, why aren't we just mutating wildly every single day? Well, it's because our cells have evolved incredibly specialized proofreading and repair teams. We're talking mechanisms like base excision repair, nucleotide excision repair, and mismatch repair that constantly scan the DNA, snipping out damaged bases and patching them right up. It is a relentless, microscopic maintenance crew working 24/7 to prevent catastrophic system failure. And even when the DNA is perfectly intact, cells don't just indiscriminately express every single gene all the time. That would be chaos. Instead, they use highly sophisticated regulatory networks. In simple bacteria, mechanisms like the LACA operon act as literal on-off switches, so genes are only active when specific food is present. In more complex organisms like humans, we use epigenetics, chemical tags like methylation that can turn genes on or off without altering the underlying genetic sequence at all, allowing our cells to perfectly adapt to their environment.
But as we wrap up this explainer, I want to leave you with this final, profound thought from the source texts. Despite all these incredible repair mechanisms striving for absolute, perfect fidelity, rare mistakes do still slip through. We call these mutations. Now, while many of them are harmful, a tiny, tiny percentage are actually beneficial. And without those rare, unrepaired mistakes, evolution completely stops.
Mutations are the ultimate source of all genetic variation. So the very errors that our cells fight so incredibly hard to prevent are the exact same errors that drove the magnificent diversity of all life on Earth. Makes you wonder, doesn't it? Is the secret to life's incredible success actually hidden right there within its flaws?